The two pathways of photosynthesis are the light reaction and the light independent cycle. During the light reaction, water is stripped of its electrons and donated to NADP, which then becomes NADPH, a very energy-rich, potential energy-rich molecule, and ATP is also generated during the light reaction. And when water is broken down or stripped of its electrons, it actually, oxygen is released as a byproduct. All of this is going to be happening in the thylakoid membrane of the chloroplast. Now again, during the light reaction, you're making ATP and NADPH, both energy-rich molecules. Those energy-rich molecules are actually used in the Calvin cycle or the light-independent cycle. So during the Calvin cycle or the light-independent cycle, what's going to happen is carbon dioxide from the atmosphere is going to be spliced together to make sugars. So this uses the ATP and NADPH from the light reaction. So the overall formula for photosynthesis is 6CO2 plus 6H2O to make C6H12O6, which is a sugar, plus 6O2. So your CO2 is spliced together to make something larger and more complex. So this is going to be an endergonic pathway. CO2 is reduced, or it's the receiver of electrons, and you create something large through an endergonic process. Now, simultaneous to this, H2O is actually being oxidized or stripped of its electrons, and as a byproduct, oxygen is released into the atmosphere. So CO2 is used from the atmosphere, so it's taken in from the atmosphere, and O2 is being released back out into the atmosphere. So to break down the light reaction into a few steps, we start off with light capture. So this whole thing, this structure right here, we call this the photosystem or a photosystem. The photosystem is composed of a light harvesting complex, or you could say the antenna complex, and then you also have what we would call a reaction center. So the reaction center, or within the reaction center, you have chlorophyll A, which should sound familiar. It's going to be that pigment, that green pigment that contributes to the green color of plants. So again, your photosystem is composed of the antenna complex as well as a reaction center. So to start off, we have light capture, and that's where a photon is absorbed by a group of light-absorbing pigments, chlorophyll A, chlorophyll B, carotenoids, or accessory pigments. And that energy is, you could say it's captured by an antenna complex. You could kind of view an antenna complex like you would view a satellite dish. It's receiving as much signal as possible by expanding its surface area and funneling all of that signal down into a central point. The same thing happens within your antenna complex. It's capturing as much light as possible of various wavelengths, and it's transferring that energy down into the reaction center. So first you have light capture. Light is captured, or you could say the light energy is captured. That energy is transferred down into the second step, which is the electrons are moved to the reaction center. So excited electrons move from all accessory pigments to chlorophyll A in the reaction center. So once that energy reaches the reaction center, or chlorophyll A kicks off an electron. So an electron is released from chlorophyll A. So here we have an electron that is released, and it meets your first electron acceptor. And then from there, we actually have a few more steps that we'll talk about. But this chlorophyll A, when it releases its electron, it must at some point replenish its electron. The way it replenishes that electron is it takes water and it oxidizes water, stripping the water of its electrons, therefore breaking apart the bonds between hydrogen and oxygen, and therefore releasing oxygen, dimolecular oxygen, out into the atmosphere. So water is broken down within the light reaction. So after that electron is released from the reaction center, water will be oxidized to replenish that electron. So water is oxidized to replenish the electron in that reaction center. So the third step, what's going to happen is that electron, much like in the electron transport chain of cellular respiration, will move from protein to protein to protein and eventually to another photosystem. We'll talk about that in a second. But as that electron moves from protein to protein to protein, much like the electron transport chain in cellular respiration, that kinetic energy of electron movement is used to drive forward these proton pumps, or a proton pump in this case. So this hydrogen moves from the stroma, so the juice of the chloroplast, into the lumen of the thylakoid. So remember those pancake-shaped flat sacs? Well, within those sacs, we're pumping hydrogens into that area. So it's going to be a very acidic environment within each of those thylakoid sacs. Much like the electron transport chain in cell respiration, you're building up a hydrogen gradient or a proton gradient. You have a high concentration of protons in this space, and when coupled with ATP synthase, well, it's going to allow, like a dam, some of this hydrogen to trickle through, and in doing so, it churns ATP synthase. It physically moves ATP synthase, allowing it to fuse ADP to inorganic phosphate to make ATP. So ATP is going to be made using the proton gradient and ATP synthase, which is later used in the light-independent reaction. So let's still look at the fate of this electron. You kick off the electron from the reaction center. It moves from protein to protein to protein, driving forward the proton pump, creating this hydrogen gradient or proton gradient. Well, it eventually reaches what we would call another photosystem. And then once it hits that photosystem, it's excited again because light isn't only making contact with one photosystem at a time, it's making contact with the entire plant. And so while this photosystem is being bombarded with light, another photosystem is also being bombarded with light. So once this electron reaches the other photosystem, what happens is that electron is kicked off because it's excited by that light energy, and it moves down another electron transport chain. Now, much like in the electron transport chain of cellular respiration or aerobic respiration, you eventually have a final electron acceptor. In this case, the final electron acceptor instead of oxygen is what we would call NADP, which is like NADH, and I like to associate the P and NADPH with photosynthesis or for photosynthesizers. NADP plus is the final electron acceptor in the light reaction of photosynthesis. So NADP plus receives electrons and then forms NADPH, an electron carrier. So if you notice here, you have photosystem 1 on the left, and you have photosystem 1, sorry. So if you notice here, you have photosystem 2 on the left, photosystem 1 on the right, and chronologically, you go from photosystem 2 to photosystem 1. I know this is kind of counterintuitive, but the reason for this is photosystem 1 was discovered first, and then photosystem 2 was discovered after that. So if I were to ask, where does photosystem 1 get its electrons from? You would say H2O, because remember, water is split. Water is oxidized, stripped of its electrons to replenish the reaction center in photosystem 2, and that electron will move down its electron transport chain to photosystem 1. And once it reaches photosystem 1, that electron from photosystem 2 is actually replenishing the electron in photosystem 1. So photosystem 1 receives its electrons from photosystem 2. Photosystem 2 receives its electrons from water. So because light is used in this light reaction to create ATP, we call this photophosphorylation because light is required for it. And so to make the ATP, or so the way ATP synthase makes ATP is it takes a adenosine diphosphate, attaches it to an inorganic phosphate to make adenosine triphosphate. So we call it photophosphorylation. So this is depicting you could say the reaction center, chlorophyll A, having its reaction center excited by light energy. And then that electron will move from protein to protein to protein to protein to protein. And in the process, making coupling its hydrogen gradient that it's generating through its electron transport chain with the enzyme ATP synthase to make adenosine triphosphate. So like I mentioned earlier, photosystem 1 was discovered first, then photosystem 2 was discovered second, and so we've named them accordingly. Electrons are going to move, however, in the chronological order from photosystem 2 to photosystem 1. So electrons move from photosystem 2 to photosystem 1's reaction center, where more photons excite another chlorophyll A. So you have photons, which activate your antenna complex, or you could say is absorbed by your antenna complex. That light energy is funneled down to your reaction center of photosystem 2. An electron is kicked off, follows an electron transport chain, and in the process pumps protons into the thylakoid lumen, creating a proton gradient. Eventually that electron that was kicked off from photosystem 2 reaches photosystem 1 to replenish the reaction center within photosystem 1. That electron within photosystem 1, or the reaction center of photosystem 1, is excited by light energy, kicked off, follows another electron transport chain, and the final electron acceptor is NADP+, forming NADPH once it receives that electron. And the enzyme that's going to catalyze this process is NADP reductase. It reduces NADP to create a charged molecule that eventually picks up a proton to form NADPH. So again, like mentioned earlier, an electron and proton are transferred to NADP+, forming NADPH. And like I mentioned earlier, NADPH, as well as ATP that you're going to be using, that you generate through ATP synthase, both of those components, NADPH as well as ATP, are going to be used in the Calvin cycle. So your photosystem 1, if you might notice this on some figures, is that photosystem 1 is usually labeled P680. That's because it mainly absorbs light at the wavelength 680 nanometers. And then oftentimes you'll probably see photosystem 1 labeled on figures as P700, and that's because it absorbs light at the wavelength 700 nanometers. So here is your photosystem 2, again, which derives its electrons from water. And then here is your photosystem 1, which derives its electrons from photosystem 2. Okay, so we just talked about all of the details of the light reaction. The reactants that go into the light reaction are going to be photons of light as well as water. So the two things that are utilized within the light reaction are photons, or light energy, as well as water. And what we're producing through the course of the light reaction is oxygen as a byproduct, NADPH, which is used in the Calvin cycle, and ATP, which is also used in the Calvin cycle. So here, this is on the left, it kind of depicts what's happening in the light reaction. Water is going in, light is going in, oxygen ends up coming out, and what we're producing is NADPH and ATP. So then we're going to move on to the light-independent reaction, or the Calvin cycle. So here's just another summary depicting all of the steps of the light reaction, starting with photosystem 2, bringing its electron to photosystem 1, photosystem 1 bringing its electron to NADP reductase, forming NADPH, and then that proton gradient created through the electron transport chain is used by ATP synthase to create ATP. Again, we're making ATP, which is pumped out into the stroma. We're making NADPH, which is brought out into the stroma, because that's where the Calvin cycle happens. So the Calvin cycle happens, it occurs in the stroma. It uses NADPH and ATP from the light reaction. So the thing about NADPH and ATP is that we do not store, or plants or photosynthesizers do not store reservoirs of ATP and NADPH. Typically, when NADPH is needed, when ATP is needed, it creates it as it's needed and it is used immediately, but it does not store ATP. ATP and NADPH are very volatile, or you could say unstable molecules. And so if they're not used immediately, they will eventually break down. what that NADPH and ATP are used for is carbon fixation, or the reduction of CO2 into a larger, more complex carbohydrate. So enzymes in the stroma will use NADPH and ATP to fix CO2 from the atmosphere. So here's a summary of the light-independent reaction. So within the Calvin cycle, the first step is going to be carbon fixation. This is where we are taking atmospheric gaseous CO2 and we're fusing it together with a larger carbon molecule or organic molecule to create some kind of solid material. So we're going from atmospheric gaseous CO2 and we're adding it to something to make it solid. So through this fusion, what we get is 3-phosphoglycerate. So here we have a 3-carbon molecule, and in this case it's depicting that we have 12 3-carbon molecules, and that makes sense because if you have 6 5-carbon molecules and you fuse that together with 6 single carbons, you're getting 6 6-carbon molecules. You break those 6-carbon molecules in half, you make 12 3-carbon molecules. So here we make 3-phosphoglycerate. Now at the end of carbon fixation, which is just taking atmospheric CO2 and fixing it to something that's solid, we move on to the reduction and sugar production steps. So reduction meaning we add electrons to it. And as you see here, it's a pretty energy intensive process. You use a single ATP per 3-phosphoglycerate to bring it to its final state, which is glyceraldehyde 3-phosphate. So we're using up a bunch of ATP through a multi-step process to create glyceraldehyde 3-phosphate. We're also using NADPH, which again has a lot of potential energy. So this, like I mentioned before, this is an endergonic process, meaning we're building something larger and complex from many smaller things. So we're using up 12 ATP, we're using up 12 NADPH, and we are creating what we would call glyceraldehyde 3-phosphate. Now, only a fraction of that glyceraldehyde 3-phosphate is fated to become sugars, whereas the remaining glyceraldehyde 3-phosphate is actually used to regenerate ribulose bisphosphate, which is what we started off with. So in this case, you could see the ratio, out of the 12 glyceraldehyde 3-phosphate we create, we're only using 2 to create sugars, and then the remaining 10, we're trying to recycle what we started off with. So in this case, you've got 6 ATP being used to regenerate ribulose bisphosphate. All right, so this is going to break it down in a little bit more detail. The 3 processes: carbon fixation, the reduction of that 3-phosphoglycerate, and sugar production, and then your regeneration of ribulose bisphosphate. So the first step, you get CO2 to combine with the 5-carbon ribulose bisphosphate. The enzyme that catalyzes this very first reaction of the Calvin cycle within photosynthesis is rubisco. So it's hypothesized that rubisco is the most abundant protein on Earth because it's likely the most abundant enzyme on Earth because we have more producers on Earth than we do consumers. So here it shows a 1, 2, 3, 4, 5-carbon molecule. You're adding a carbon to it, making it a 6-carbon molecule. Then you break it down into 2 3-carbon molecules, each containing a phosphate group. So next, what we do is we take that 3-phosphoglycerate that we created during carbon fixation, and we reduce it to glyceraldehyde 3-phosphate, still a 3-carbon molecule. But this is the good stuff. This is the stuff that we're going to use to make sugars. If you take 2 3-carbon molecules and you splice those together, you can make a 6-carbon sugar easy. And again, this process is going to be energy intensive. It's using ATP and it's using NADPH. You're adding electrons to it, you're adding phosphates to it. And then lastly, again, we are regenerating ribulose bisphosphate using ATP on the remaining glyceraldehyde 3-phosphate. So a small fraction of that glyceraldehyde 3-phosphate is actually fated to become a sugar, whereas the rest is used to regenerate that starting material, ribulose bisphosphate. And again, an energy-intensive process. We're using up ATP to make that happen. So the end product of the Calvin cycle is technically glyceraldehyde 3-phosphate, a 3-carbon molecule. And that glyceraldehyde 3-phosphate is then used to create sugars. Some of this sugar is stored as starch, and then some of the sugar is used to make sucrose, which we know sucrose is going to be a combination of glucose and fructose, two isomers of one another. So the summary of the Calvin cycle, the reactants going into the Calvin cycle are going to be NADPH from your light reaction, ATP from your light reaction, and CO2 from the atmosphere. And the products are going to be glyceraldehyde 3-phosphate, which is eventually converted into C6H12O6, which could be glucose, which could be fructose. Remember, those are going to be isomers of one another, meaning they have the same chemical formula. However, they might have a different arrangement of atoms.